Thank you, Donnie. Now we proceed. We will have some question at the end, if Art keeps on time.
I'll try. You'll remind me, I'm sure, if I don't.
Okay.
Thanks very much, Luciano. Thanks very much, Greg, for a terrific talk and actually a very good introduction to what I'll be talking about. And that's how ventilator-induced lung injury might lead to the development of multisystem organ failure. There are a number of mechanisms by which mechanical ventilation could lead to multisystem organ failure. Certainly we know that there's effects of mechanical ventilation on oxygen delivery. It's been known for at least 50 plus years that increases in airway pressure lead to changes in pleural pressure, which affect preload and afterload and could have marked hemodynamic effects. Patients can be hypoxic, severely hypoxic, which again can lead to problems. The endotracheal tube we use, multiple antibiotics, can all lead to nosocomial infections, which can again be a presage of multisystem organ failure. We have various interventions, fluids, inotropes, etc., which again can have detrimental effects. And finally, there can be the direct effect of the lung and the mechanical ventilator as a system, and the lung being a source of an inflammatory response that could potentially lead to multisystem organ failure. And what I'm going to focus on the next 20 minutes is this topic right here. That is the interaction of the ventilator and the lung leading to an inflammatory response. Now, the lung/mechanical ventilator complex can be a source of systemic inflammation. And it's actually an interesting the lung itself is an interesting source and an interesting possibility for this. The lung is unique because it receives virtually the entire systemic circulation. It has a huge vascular bed, and it contains about 20 to 25% of marginated neutrophils. So anything that happens to the lung, there's immediately a source of neutrophils which can be activated by various mechanisms, as Greg discussed. In addition, the lung has a huge surface area and can be the portal of entry for many pathogens which enter through the lung, can reach the distal lung, and be activated. It's also a very metabolically active organ, with the epithelium and the endothelium producing many substances, many molecules, and I'll spend a little bit of time, actually just one slide, showing an example of this and its relevance to ventilator-induced lung injury. So you can see the lung really is a perfect source, if you like, to be a motor of systemic inflammation. So a hypothesis I'd like to develop over the next few minutes is that, one, mechanical ventilation can cause release of mediators in the lung, then that these mediators can leak from the lungs into the systemic circulation, as hinted at by Greg in a previous talk, and finally that mediators can lead to end organ dysfunction. And I'll present data on each one of these individual topics to at least present a hypothesis and supporting evidence that this hypothesis may be correct. What about the fact that mechanical ventilation can lead to release of cytokines and other inflammatory mediators? We published a study a number of years ago. Lorraine Tremblay was the first author on this, where we took an ex vivo lung model, took lungs out of rats, ventilated them in non-perfused lungs, ventilated for 2 hours, using 4 different ventilatory strategies. Strategies were either a control group, a small amount of PEEP, 3 centimeters of water, tidal volume of 7 mils per kilogram. We then used another group with PEEP of 10 centimeters of water, tidal volume of 15 ml. This led to over-distension. Medium volume, 0 PEEP, same tidal volume, the same change in volume with time, now 0 PEEP. And finally, a group that had both over-distension and 0 PEEP. And remember, these are ex vivo lungs, so at end expiration, the lungs collapse completely, magnifying the kind of injury, magnifying the Adler trauma. We then waited a couple of hours, as I said, and made a number of measurements. One of the things we did was do a bronchoalveolar lavage and measured various cytokines. And here on this slide, I'm showing you one of the cytokines, tumor necrosis factor alpha, a key mediator in the sepsis cascade. You can see the control values are down here. The actual number doesn't matter much. It's the relative change that's important. With medium volume high PEEP, So we overdistend the lung. You can see there's roughly a tripling of TNF levels. When we used medium volume 0 PEEP, so now there was much less overdistension, but now 0 PEEP, a lot of atelic trauma, there was a further doubling. This was about a 6-fold increase over control in TNF. And when we combine both injuries, that is overdistension and allowing the lung to be de-recruited, there's a marked increase. There was a 56-fold increase in TNF simply in mechanical ventilation for 2 hours in this ex vivo model. We also looked at a number of other cytokines, and these are shown on the next slide. Pro-inflammatory cytokines, a chemokine, that's a cytokine that attracts neutrophils into the lung, similar to IL-8 in humans, and an anti-inflammatory cytokine. And you can see there's some differences in the various cytokines, but in all of them there's the same pattern, with the groups on the right causing more of a release of these various cytokines. So this could be one of the mechanisms by which neutrophils might be recruited into the lung. The lung mechanical ventilation might overdistend lung, cause de-recruitment, and then cause neutrophils to be called back into the lung. We also looked at, and published recently, just a couple of months ago in Critical Care Medicine, what cell types might be involved in producing these cytokines and releasing these cytokines. And in fact, we originally thought that the neutrophil or alveolar macrophages would be the prime cell that's turned on. It turns out that based on various studies looking at immunohistochemistry, I'll show you in a minute, in situ hybridization for TNF, that the epithelial lining cells of the lung are actually a major cell type that produces the cytokines. That makes sense in a way. You think of the lung, if you like, feels the entire mechanical ventilation and is stretched with each breath. That was the in situ hybridization, also immunohistochemical staining for TNF protein, showing it's largely in the epithelial cells. So these data suggest that the epithelial cells are important, not just as with reduction in barrier function during ventilator-induced lung injury, but actually might be a source of some of the cytokines which we were discussing. So the next question is, if the lung can release various mediators in response to ventilator-induced lung injury, can these mediators leak from the lungs into the circulation, as Gregorio mentioned? Well, there's a number of studies that have addressed this, both ex vivo, the perfused models, in vivo models, and actually human data. I'll just take one slide from Stefan Uhlig's group, again from von Bethmann, published a few years ago. They used an ex vivo mouse lung model that's perfused, and they could then measure in the perfusate distal to the lung, the arterial side, if you like, various cytokines. And here is time on the x-axis. They start high overventilation at this point here, and you can see under control conditions, TNF remains relatively low. With high peak inspiratory pressures, TNF levels in the perfusate increase dramatically, showing that mechanical ventilation by itself can lead to release of TNF, not only in the lung itself, in the air spaces, but also in the blood perfusing the air spaces. So there's evidence that mechanical ventilation can lead to cytokines, which can get into the systemic circulation. And I said there's animal data, there's also human data. This actually is the same slide that Greg showed you earlier. Again, in this study published in JAMA a couple of years ago, you can see that the blood levels, these are serum cytokine levels of IL-6, increased in the control group, decreased in the minimal stress group. This study per se didn't prove that those cytokines came from the lung, but there's other data from a couple of other groups that looked at cytokine levels across the lung, showing there can be an increase in cytokine levels across the lung. That is, the lung releases the cytokines into the circulation. The final question I'd like to address is whether mediators that are released from the lung, can they have an effect on end organ dysfunction? Because in the introduction I pointed out that most patients who are being ventilated with ARDS don't die of hypoxia, they die of end organ dysfunction. And it's been a bit of a puzzle for many years why that's the case. We looked at this question. Yumiko Amai, who's a fellow who spent some time in the lab, who's currently in the lab as well from Japan, did a very nice study trying to examine the systemic effects of ventilator-induced lung injury. And the model she used was that of anesthetized rats given intratracheal injection of hydrochloric acid. A model we've used previously, I think a reasonably good model simulating acid aspiration in humans. The animals were then randomized into groups, either low tidal volume, high PEEP, call that protective strategy, or high tidal volume, zero PEEP, non-protective strategy. And we made a number of measurements, including hemodynamics, various enzymes, cytokines, and most importantly for this talk, made measures of apoptosis. Because of time commitments, I'm not going to show you the hemodynamic data. Trust me that for these groups, hemodynamics were virtually constant. I think an important part of the protocol, because if there were marked differences in hemodynamics, they could explain the findings. What about implications for novel therapeutic approaches? Again, Greg has sort of hinted at this in terms of anti-inflammatory approaches, as well as genetic prediction of who might be a candidate for development of ventilator-induced lung injury. One approach that might be useful is something called the heat shock or the stress response. This is, it's been known for a number of years that if one increases temperature by just a few degrees centigrade, this leads to suppression of translation and transcription of most genes. But there's an increase in expression of a group of genes which are called the heat shock genes or the stress response genes. These genes are important because they have been shown to be cytoprotective. Various models have been used to look at this. In fact, Jesus Villar published a number of studies looking at the protective effect of heat shock proteins in sepsis. We wondered whether the heat shock response might be protective in ventilator-induced lung injury. Sergio Ribeiro was the first author. This was published last year. So we took heat or sham heated rats, excised the lungs 18 hours later. It takes time for the heat shock response to develop, and then ventilated them with relatively large tidal volumes. We measured pressure volume curves, cytokines, and a number of other factors. The results were, in terms of lung compliance, the sham group had a decrease in lung compliance with this model of 45%, whereas the heated group had a decrease of only 18%. This was significant. When we looked at cytokine levels, we've measured a few cytokine levels. I'm only showing you TNF here. You can see the control animals, there was an increase in TNF after ventilation. The heated animals, those heated 18 hours earlier, so the lungs were the same temperature during ventilation, a marked decrease, suggesting that this approach might be useful to abrogate ventilator-induced lung injury. How this might be used might be in the context of gene therapy, perhaps, or other approaches. There's a recent article in Journal of Clinical Investigation by Yoram Wise and colleagues using the heat shock response in gene therapy to abrogate cecal ligation perforation, ARDS. Another approach might be to use anti-TNF antibodies, and this is a study published a few years ago by Yumiko Amai. This model is a rabbit bronchoalveolar lavage. The animals are lavaged, actually lung lavage, produce hypoxia, PO2 of about 100. And you can see that under control conditions or with the control antibody, oxygenation is pretty poor. They then used an antibody to TNF and found that at low dose and at higher doses, that this antibody to TNF, now attacking the inflammatory response, or attacking TNF, had an increase in oxygenation. So there's at least a possibility that we might be able to affect oxygenation and ventilator-induced lung injury with TNF antibodies. Conclusions then are, I think at the bench, we have very strong data from many, many models that mechanical ventilation can augment lung injury and specifically can lead to an increased inflammatory response. There's data at the bedside that these protective ventilatory strategies can decrease cytokine levels, can decrease organ failure, and most importantly, can decrease mortality. And finally, what does the future hold? I don't know, but it's possible. It's possible that in addition to ventilatory strategies, we might have novel anti-inflammatory strategies to minimize ventilator-induced lung injury. Thank you very much.